Hello, welcome to another video on genetics. In this video, we will learn about the law of independent assortment. This was the outcome of Mendel's work on his hybridization experiment with bees. Now, we have seen in our previous video about the monohybrid cross experiment that he conducted in great details and came out with certain outcomes. Now, Mendel did not stop there, rather, he moved on to something which is known as the dihybrid cross experiment. And the dihybrid cross is a cross made between two organisms, keeping in mind observation of two traits. It can be any two traits, and then looking for the outcomes. Now, breeders and geneticists perform these kind of dihybrid cross experiments or procedures for various purposes. Similarly, we can have a trihybrid cross or a tetrahybrid cross that we have already spoken about in our previous videos and some other videos. Now, Mendel had chosen 7 different characteristics, and for his dihybrid cross experiments, he chose different pair of traits and studied them in great detail. So I'm only going to illustrate one of those experiments here, one of those combinations for the dihybrid cross. So in this particular experiment, he chose the seed of the pea plant for observation, and the two characteristics that he chose was the texture of the seed and the color of the seed. Now the seed has two textures, either it is smooth or it has a wrinkled surface. Also, the seed can either be green in color or yellow in color. Now he crossed the plant keeping in mind observing the change in these two characteristics after the hybridization. Now the beginning of the experiment, the parents that are chosen are the same as in a monohybrid cross, which means that the parents are pure lines. So one of the parent was a pure line for the dominating conditions, round and yellow, round being the dominating condition. That is a homozygous condition for both the traits in this case. And the other pure line that he chose was a plant which was wrinkled green, and both of these traits are recessive, and they can only be recessively expressed. The phenotype can only be recessively expressed if they are in the homozygous condition. Now, these two plants, if they have to hybridize, they will produce gametes, right? Now, what are the possible combinations? Well, the possible combination is this R can combine with this Y, and we get one possible combination here. This R can also combine with the other Y, which is more or less the same thing. So we have one combination for this particular gamete. If you look at the other other allele, I'm sorry, if you look at the other allele of the allelic partner, it can combine with this Y and enter into the gamete, or this R can combine with this Y and enter into the gamete. So we have probably two possible combinations right here. Similarly, in the case of the wrinkled green plant, during the process of gametogenesis, what are the possible combinations available? So this R can combine with this Y, or this R can combine with this Y, and the possible outcome that we can have is small r, small y. Likewise, the second allelic partner of the allelic pair here, RR, can combine with either this Y or it can combine with this Y, and it can give rise to RY. So we have got two possible combinations here also. Now, when Mendel crossed these two parents, round and yellow plants that had round and yellow seeds with plants that have wrinkled green seeds, he found out that the progeny, the entire first generation of offsprings, all the seeds were round. So just recall the monohybrid cross experiment. What happened in the monohybrid cross experiment? In the monohybrid cross experiment, all the recessive traits had vanished. They had disappeared in the first generation of offsprings, and that is observed even here. The dominating traits, which is the round and yellow, is expressed in the first generation, and the recessive traits, which is wrinkled and green, are absolutely absent here. Now let us draw a Punnett square for this condition, and let's see what is the outcome. So we have two combinations, possible combinations from our first parent, that for the genotype of the gamete, and we have got two possible combinations for the genotype from the other parent. And here is the Punnett square for this condition. So we've got capital R, capital Y, capital R, capital Y on the top, and small r, small y, small r, small y on the side. And the combination then becomes a heterozygous condition. So we get 4 heterozygous conditions. Capital R gives us this. these two combinations, capital R, small r. Then we've got capital Y and small y. And likewise in the other squares, if you see, if you fill them, capital R and small r will give us capital R, small r. And capital Y from here and small y from here, capital Y, small y. Likewise the other. So we get four possible outcomes, and all the four possible outcomes have the same genotype, which is a heterozygous condition for both the allelic partners or allelic pairs for two different traits. The outcome for the phenotypic outcome, which is here, the phenotype of the first generation is all round yellow, 100% round yellow seeds. The wrinkled green are absolutely missing. And what is the genotype of all these seeds or the offsprings? It is a heterozygous condition, capital R, capital Y, small y. Now, the result is more or less nearly the same as, or basically the same as that of a monohybrid cross. Now, Mendel again did not stop here, and he selfed the first generation offspring. So the genotype as capital R, capital Y, small y were selfed. These plants were selfed. And now let us first of all look what are the possible combination of gametes that can be formed or that are formed by the offsprings of the first generation. So let us have a look. So capital R can combine with capital Y, and we get this combination capital R, capital Y. Capital R can also combine with small y to enter into the gametes, capital R, small y. Small r can combine with capital Y. These two different allelic partners of two different traits can also combine, small r's, capital Y. And small r can also combine with small y, forming the fourth pair, small r, small y. I hope you know why am I making these combinations, because from each allelic pair, one allelic partner is separated during the formation of gametes and it enters the gametes. So capital R enters the gamete. Likewise, from the other allelic pair, one allelic partner can enter the gamete. It can either be capital Y or small y, and that is what we are doing here. So we get four possible combinations for this reason. This is called the law of segregation that we've studied. So based on law of segregation, we get four possible combination of these two traits in the gametes. So we get four possible types of genotypes of gametes. Now, if we build up, because the plant has now been selfed, let us see what is the combination. What happens when the gametes from one plant fertilizes the gamete of the other plant. So let us build a Punnett square, and this Punnett square, have a look at this Punnett square is fairly larger. Why is this larger? Because there are four possible combination of gametes possible: capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. And because it's a selfing, so on the other side also we'll have the same combination of gametes or the genotype of gametes. And when we now fill that, let's start filling the Punnett square. And on filling the Punnett square, we get capital R, capital Y, capital Y. Have a look on the top and the side. And then let us combine this one, the second one, with the first one here, and we get capital R, capital Y, small y, and likewise all the others are filled, and I get the particular genotypic chart here. And let me just read it out to you, so that I want you to read it out for yourself and just figure out what are the probable combinations: capital R, capital Y, capital Y, capital Y, capital R, capital Y, small y, and so on and so forth. So we get around 16 possible combinations or outcomes. Now, what will be the phenotype of the offsprings and the genotype of the offsprings? Is that what we are interested in? So what is the phenotype of the first one? Let's have a look at it. So this is going to be a round and a yellow seed. So first genotype that is possible is round and yellow. So I've written the first phenotype that is possible. It is round and yellow. Let's look at the second one. This is again round and yellow because homozygous dominating condition, and in the second allelic pair we've got a heterozygous dominating condition, capital Y. So round yellow seeds again. In this third case, again round yellow seeds. Fourth case, round yellow seeds. Fifth case, round yellow seeds. Sixth case, round and green. So this is the second phenotype. So we get the second type of phenotype, which is called the round and green seeds are now seen. Again, round and yellow, we've already done this, and round and green. So we get more round and greens now coming up, and round and yellow. Was this combination there? Yes, we had this combination here, round and yellow. Now we are going to get round and yellow. So we get, yes, we've done round and yellow, and wrinkled yellow. Now, this is the third combination. Look at this. The third possible phenotypic combination is wrinkled yellow, the outcome, wrinkled yellow. And this is again wrinkled yellow. So you see wrinkled yellow. And this is round and yellow. We've already done round and yellow and round and green. Now, this is, we've already done this, yes. And then we got wrinkled yellow. We've done this. And finally, we got wrinkled green. And here is the fourth possible combination. So we have four different phenotypes that are formed in the second generation or the F2 generation. So four phenotypic combinations are possible. But what about their genotypic ratio? What about the genotypes? How many round yellows do we have? Let us count the round yellows, and let's use a black color to strike them off. So let us first of all count all the round yellows. And we got round yellow 1, we got round yellow 2, we got round yellow 3, 4, 5, not this one, 6, 7, and this is 8, and this is 9, and we get 9 round yellows. So let's just write 9 here. And how many round greens do we have now? Let us look at round green. So this is round green 1, round green. This is also round green 2. And let's look at, so this is round green 3 down here. Any more? No. So we got 3 round greens. And how many wrinkled yellows we have now? Now this is a wrinkled yellow here. And then this is also a wrinkled yellow. And then this is also a wrinkled yellow. So we got 3 wrinkled yellows, right? And how many wrinkled greens we have? Now everything else has been struck off and we only have this final one. So we put this like this and we get 1 wrinkled green. Now I've already done this. So what is the genotypic ratio? Or sorry, what is the phenotypic ratio, not the genotypic ratio? The genotypic ratio is even far more complicated. We are looking at what is the phenotypic ratio of the F2 generation. So there were 4 possible phenotypes: round green, round yellow, round green, wrinkled yellow, wrinkled green. And the ratio, phenotypic ratio, is 9 round yellows to 3 round greens to 3 wrinkled yellows to 1 wrinkled green. So this is the classic 9:3:3:1 ratio. Oh, what does this mean? Well, at the moment, it means nothing. So now, what Mendel did, he moved a step ahead and he said, well, what is this? What does this number? What does this number? Does it make any sense? Does it have any particular meaning? So what this person, Mendel, did, he chose the same trait for comparison. So what he said was, let me just see what is the ratio of texture in a dihybrid cross and what is the ratio of the contrasting characters for the color in this particular dihybrid cross experiment. So what he meant to say was, what is the ratio of round to the wrinkled seeds? What is the ratio of round to wrinkled seeds? And what is the ratio of the yellow color to green color in the F2 generation of the dihybrid cross experiment? So let us just count. Let us see how many round seeds in total do we have. So we have like a total of 16 possible combinations. Out of these 16 combinations, how many combinations have round seeds? So let's look at this. So here we got round seeds. How many round seeds do we have? We have got like 9 round seeds. And how many round seeds do we have in round and green? We get 3. And do we have any other round seeds? No. So a total of 9 plus 3, we got 12 round combinations. So let's write 12 round combinations here. And how many wrinkled combinations do we have? So we see, look at this. So we have got wrinkled here. And how many wrinkled do we have in wrinkled yellow? We got like 3 seeds that are wrinkled, right? So, and how many wrinkled do we have? Do we have any more wrinkled seeds? Yes, we have wrinkled seeds in this combination, wrinkled green. And we have got 1 wrinkled seed from here. So, but not seed, basically a combination. So we get 4 combinations of wrinkled seeds. Now, how many yellow seeds do we have in these 16 combinations? And we see we have got 9 yellow seeds from here and you get 3 yellow seeds from here. So 9 plus 3 is 12. There are no more yellow seeds. So we got like 12 yellow seeds. And how many green seeds now do we have? We got like 3 green seeds here and we got 1 green seed here, and this makes it like a 4 green seed. Now, if you look at this particular phenotypic combination of texture and color, let us have a look at this. If you simplify this particular ratio, the ratio of texture. So here is texture. Let us only look at the texture again. So the ratio of texture, which is round to wrinkled, 12 is to 4, if you simplify it, you simply get it as 3 is to 1, which is 4. 12 divided by 4 is 3. So 3 is to 1. The ratio is 3 is to 1. And what is the ratio here for color? Yellow is to green is, again, if you simplify it, you get is 3 is to 1. So just recall what was the ratio at the end of the monohybrid experiment. Wasn't it the ratio? Isn't the ratio the same as that before, the phenotypic ratio? 3 is to 1. What does this mean? Now, this will make certain sense. And the sense is that the allelic pair for two different characteristics separate from each other during gamete formation independent of each other. They do not alter the separation of the genes that are controlling the traits for a different character. What do I mean by that is, let me just write it down here. So we had RRYY in the as the parents and RRYY. This outcome here at the end tells us, see, because this outcome is the same as the one seen in the monohybrid cross experiment during the law of segregation, right? And what does law of segregation says? It simply says that from each allelic pair, let us say from each allelic pair, one of the partner will move into one of the gametes. And uh, anyways, let's just extend this to the example that we are doing now. Now, the outcome of this particular experiment here is, and this is what Mendel concluded, is that during gamete formation, each allelic pair acts independent of the other. This means Y, capital Y will not exert any influence on capital R when gametes are being formed. These two allelic pairs will separate out and move into one gamete each. So this will go here, this will go here. And these two allelic pairs, capital R, will not influence which of the two will have to go where. They will independently assort themselves. This can go here or this can go there. So this is called law of independent assortment, right? And the statement for law of independent assortment is that the alleles for a character assort independent of alleles for other characters. This means that the allelic pair, one allelic pair for a character will not influence the allelic pair of the for the other character during the process of gametogenesis or formation of gametes. And well, this particular ratio, 3 is to 1 at the end, the phenotypic simplified ratio, actually points out in this particular direction. I hope you've understood what the second law of, what Mendel's second law means, the law of independent assortment. I will see you in some other video in the future. Thank you.